welcome to the Biological Life Sciences channel. Today in this video we are going to see the semi-conservative replication of DNA and how the DNA is expressed or how the DNA is replicated, that we are going to see here. Okay, so the main point here is the replication of the DNA. You know that the parent a cell divides and forms two different cells. So when the cell is dividing, it is forming two different nucleic acids and two cells are getting different copies. For example, this is the parent cell having certain DNA, so it has to replicate its DNA in two different parts or two equal parts. Okay, so here the two cells are going to get the equal amount of DNA. So what is the mode of replication in these two cells, that we are going to see in today's video. Okay, so first of all, let me explain you the first types of replication or different modes of replication. Then we are going to see how semi-conservative mode of replication of DNA was studied. Okay, so first of all, the first replication of DNA is called as the conservative replication of DNA. What happens in this conservative replication? Suppose this is the parent DNA. This is the parent DNA. Then the offspring DNA will have a new copy of DNA and the parent copy of the DNA will be conserved. Okay, so this is the conservative mode. So the parent DNA is conserved into one organism and a whole entire set of new DNA is formed. This is called the conservative mode of replication. Now let's see what is the semi-conservative mode of replication. The semi-conservative mode of replication, here you can see this is the parent DNA, suppose. Then the two organisms that are formed here will have each strand of the parent DNA and a new strand of the offspring will be formed. Means in this new two offsprings that are formed here, you can see one DNA strand will be newly synthesized, that is green color here, and the other DNA strand, that is red colored here, will be the parent DNA. So half copy of the parent DNA is retained. So this mode of replication is called as a semi-conservative mode. Okay, this is called semi-conservative mode of replication. Okay, so now let's see the third type of the DNA replication. Suppose this is the parent DNA, and in this type of replication, what happens? The parts of DNA gets replicated in different manner. You can see here the new organism that is being formed here will have two different strands having mixture of red and green type of DNA. Okay, so it will be like this. It will be like this. Okay, so here half of the DNA, not exactly half, but in parts. The DNA replication will be in parts which will have both types of DNA. So this mode of replication of DNA is called as a dispersive mode. This is called as a dispersive mode. Okay, so the DNA that is present in all organisms, it replicates semi-conservatively. That was first time shown by Meselson and Francis-Stahl. So how they did that we are going to see in this experiment. So now for this experiment, that is also called as a Meselson and Stahl experiment, they used an organism that is very simple organism, that is E. coli. Okay, so now we all know that the DNA is having nitrogen in its structure, and the nitrogen is present in quite a bit quantity. Okay, so normally nitrogen is having an isotope, that is N14, that is quite prevalent in the nature. So this N14 isotope is quite prevalent, means what? It is having lots of percentage, and this is the normal isotope. Another isotope of nitrogen is N15, that is having one extra neutron. Due to this nitrogen is much heavier than this nitrogen. Okay, so we know that the nitrogen is present in the DNA structure. It is present as a nitrogenous base. Okay, so what they did, this Meselson and Francis-Stahl, what these both scientists did, they took E. coli as their experimental organism. Okay, so E. coli is a very simple bacterium. It replicates within the 20 minutes of its lifetime. So therefore they selected this organism. What they did first, they grew this E. coli on N15 containing medium. Okay, and they grew it for sometimes for some generations. When they did this, the whole lot of DNA from this E. coli was made up of N15 nitrogen. Okay, so now the entire DNA set present in this E. coli is having the N15 type of nitrogen, that is heavy isotope. What they did, they extracted the DNA from it, and performed CsCl2, that is cesium chloride density gradient centrifugation. They performed cesium chloride density gradient centrifugation. Okay, so when they performed this centrifugation, this DNA was having the density equivalent to the cesium chloride salt that was taken in that, the centrifuge solution. Okay, so now it was clear that the DNA obtained from this E. coli was a bit heavier than the regular E. coli, and it formed a band below the centrifuge tube, or it formed the band at a lower point in the centrifuge tube. Okay, so what they did now in the next step, they grew this same E. coli onto the normal N14 medium. Now the N15 that was used here was used in the form of a salt that is called ammonium chloride. So now this is a nitrogen source in the medium. So what they did, they used this nitrogen as nitrogen 15, and here they used nitrogen 14 in the next generation, and they grew the E. coli having total N15 nitrogen into this medium, that was normal medium. Okay, they called this medium as normal medium because it had the regular one, nitrogen 14 isotope of nitrogen in the salt ammonium chloride. Now in the medium above it was nitrogen 15. So due to this, what happened, this E. coli was having the entire set of N15 DNA. Yes, so now what happened, when they grew the E. coli into the normal nitrogen source medium, that is the N14 nitrogen ammonium chloride, what happened, the DNA got synthesized using this. Yes, so when the first generation of the cell was formed after this, first generation of N15 E. coli when transferred onto this medium was formed, it was found that it was having an intermediate DNA. Means it was having in its DNA, it was having N14 nitrogen also. Similarly, it was having N15 nitrogen also. Okay, so when the synthesis went on for some few generations, after few generations, say 5 or 6 generation, the DNA was completely turned to N14 nitrogen. It showed that the nitrogen taken up from this medium was also used in the DNA synthesis. Now let's see the experimental result in a proper manner, which will show us how the actual bands looked into the centrifuge tube, that is the cesium chloride density gradient centrifugation. Remember, this cesium chloride density gradient centrifugation is a highly specific centrifugation where you can find a difference of a single nucleotide also in the centrifuge tube. So that's we are going to see here. So first of all, let me draw the first centrifuge tube. You can see here, this is the second one, and this is the third one. Okay, so now what happened, the heavy N15 nitrogen was forming a band at this point in the centrifuge tube. Okay, so this is a N15 DNA band, and it was considered as a heavy band because all the cells was grown on N15 medium or N15 media. Now when these cells was transferred to N14 media, the cell already had N15 DNA into their cells, so it was forming a intermediate band here. You can see here, it was forming an intermediate band at this position in between, like this. Okay, so this was N15 N14 band. Okay, and now when the DNA band was extracted from regular E. coli, that is the organism used here, it formed a band like this here at this position. So the band formed was with normal and nitrogen 14. Okay, so you can see when you compare this, the intermediate bond was band was formed at this position in the center of the centrifuge tube. This suggested that the organism at this point had equal amount of DNA from parents. Okay, so what if the two cells are found from parents, then the two cells will have the half amount of DNA from the parent. Each cell will have half amount of DNA from the parent, and new DNA strand was synthesized in the both organisms, and this suggested that the DNA was replicating semi-conservatively. To prove it further, they did the growth of this E. coli for further generations. First of all, the reading was 50%. This generation, when it was grown, you got 50% of either of the DNA. Then when they moved for second generation of the E. coli on normal nitrogen 14 containing medium, they got the ratio as 75% of N15 and 25% of this normal isotope. And then when they grew it for further, then it reduced to 85.5 to 14.5, and then it was 100% of the normal nitrogen, which was continuously growing on the normal nitrogen containing media. So this was all about the semi-conservative replication of DNA. I hope you have understood this whole experiment. If any doubt, you can comment below, and if you like the video, please share the channel and comment your questions. Thank you so much for watching.